Thank you. Honored to be here.
Yeah, welcome, Dr. Pete.
Very excited to be here.
Yeah, okay, let's start. So,
Okay.
myself, Dr. Jaspreet, member and a certified metabolic health coach from dlife.in, which is India's first and the only award-winning low-carb platform. DLife was founded in 2014 by Anup Singh. Anup is a silver medalist from IIT Roorkee, and he is a recipient of the Icons of India Award. Anup is a pioneer of the low-carb movement in India with his path-breaking protocol of 100-gram carbs per day max that made lchf possible for even Indian vegetarians. In 2021, DLife launched its diploma course, which is the only course approved under the NSDC Partner, the Government of India in this segment, and the only course with the global biggest 144-hour CPD Hrs credit. So now, coming to my esteemed guest, so let me take a pleasure to introduce you to our audience. So Dr. Pete Delnoy, he is a PhD in biochemist and with over 28 years of teaching experience and now has turned the keto coach. His personal health journey began when he was diagnosed with the pre-diabetes and gout. After struggling with the traditional medical advice, he turned to the ketogenic diet, which led significant improvements in his health. Dr. Pete now runs a coaching company, Ketonic Revolution, aimed at helping others reverse the chronic disease through the dietary interventions. He is highly qualified and he is also a nutrition network coach practitioner. So for this session of the Metabolic Health Conference, we are going to talk all about the uric acid and clarify many misconceptions surrounding it. So while uric acid is often viewed as a biomarker for the gout or arthritis, its metabolic effect on metabolic processes are much broader. So we are fortunate to have with us Professor Pete Delnoy, a leading expert in the implications of uric acid for the metabolic health. So please join us for the discussion. So Dr. Pete, welcome again to the Metabolic Health Conference, Season 3.
Thank you very much. I'm super honored to be here, and let's get started.
Yeah. So my first question to you, so to start with, I would like you to come up with your personal journey and explore what made you interested in the field of health and nutrition as a biochemist.
So 7 years ago, I'm in my seventh year. I was diagnosed as being pre-diabetic, and before that, with gout.
Mhm.
And the pre-diabetes is the thing that really scared me because basically what my doctor was telling me was that this is just progressive, and that I would be type 2 diabetic down the road, but that they had really good drugs for this. But I am a biochemist, and I did research before I went into teaching, and I understood at the time that prediabetes and the gout were metabolic. And as such, they should be responsive to basically what you eat. And I do want to qualify this because we know that there are the other factors that are involved in this, exercise, muscle mass, and all of those other factors, sleep, and so on down the road. But by far, the, I think you can argue that the biggest factor here is the aspect of metabolics. So I struggled and did what the doctors asked me to do for a while, but I saw that my fasting glucose and A1C were rising. And eventually, to make a long story short, because I know we don't have a lot of time,
Yeah.
I found my way to the low carb environment, and the early studies that were published by Verda, and reversed my prediabetes in a very short time frame. But one of the lagging questions in this was the fact that I was hyperuricemic. So I had high uric acid. And in the process of going to the low carb lifestyle, I saw that condition rose significantly. And then it did fall with keto adaptation, but it only falls to the hyperuricemic level. And as you already told our audience, I have a great deal of coaching experience then, which then has ensued with this. And I know that when people that are hyperuricemic to start with go on a ketogenic diet or they're carnivore, once they get to how do I want to say this? An established metabolism. So for someone who's on a ketogenic diet, they become keto adapted. This takes, with especially older individuals, this takes months before that happens. And the carnivore diet is a little bit different because these are, in fact, two different physiological states. They're related. There's a lot of overlap, and they are completely distinct from the standard American diet physiologically.
Mhm.
But the hyperuricemia remained, and I think, as you know, there's a lot of influencers involved in this space that are asking the question, is hyperuricemia safe
Mhm.
on a ketogenic diet and so on? And so uh, as a scientist, I went into the literature in the early days, 7 years ago, basically, uh, to look at what I could find in regard to the science.
Mhm.
And as I think everyone in this space does, when you first are confronted with the low carb environment, you're like, okay, people are saying if I do this, I can reverse my disease. And they read stuff like from Gary Taubes and Nina Teicholz. But I'm talking about going a lot deeper here because for
Mhm.
me, I had two questions at the very beginning of this. And one of those questions was, what is the biochemistry that's actually driving these diseases? And as a trained biochemist, I understood that if you make a list of all the chronic metabolic diseases that we're suffering from, obesity, diabetes, the gout situation, hyperuricemia, chronic kidney disease, yada, yada, down the road you go, there's a tendency to assign a biochemical mechanism to each and every one of those, which is when trying to understand a biological function is the first mistake that people make when they start to study the science, right? Is to invent 30 different hypotheses for 30 different uh, illnesses. And I understood that if we're dealing with something that is metabolically, has a metabolic origin here,
Mhm.
that there can only be a few central biochemical mechanisms that are leading to all of this. It's just the nuances with how this happens.
Mhm.
So I started looking at gout specifically to answer this question that I was getting a barrage on my YouTube channel, and in meetings, you hear this coming up. I have hyperuricemia. I suffer from gout. Is it safe for me to be on the ketogenic diet? That's where this started. And uric acid took me to basically Rick Johnson's work, and to be fair, his collaborators, because he works with a team of people, and those guys have literally published hundreds of publications. And I started looking at uric acid, and I learned right away that number one, the fructose uric acid pathway is a central driver of all metabolic diseases. I've learned this. There's different, when you look at a bell curve of individuals,
Mhm.
and they have, different metabolic diseases, one of the first things that you see is that there's a tremendous amount of overlap. Um, Somebody who's obese, it's usually rare that they're not suffering from something else that's related,
Mhm.
right? For example, gout sufferers, 74% of them also are hypertensive, right? They have either their borderline high blood pressure or they have high blood pressure. 71% suffer from some level of chronic kidney disease, which, by the way, is because of fructose uric acid biochemistry in the kidney. All right. And I learned right away that because of the polyol pathway, which is found in all human tissue where it has rigorously been looked at, and because we're low on time, I'm just going to blurt out the line for this. Fructose uric acid metabolism is as dependent on glucose as it is fructose. And we just have published a major paper in this area that I'm lead author on in the Journal of Biochemical, in the Biochemical Journal out of the UK. And this is going to be in whatever the next issue is. I just got the proofs for it yesterday, but that article was a direct response, basically, after I had produced a video in this area. It was a direct response to a video I saw a couple summers ago, where one of the lead scientists in the carbohydrate insulin model of obesity, he was asked during that presentation, what is the role of fructose in this, in driving obesity? And he said, well, there isn't any because fructose doesn't stimulate insulin. And I fell out of my seat. And the reason I fell out of my seat when I heard that is because I know full well that opening fructose uric acid metabolism, which I believe is going on in the chondrocyte and is a central driver therefore of gout, depends as much on the glucose as it does on fructose.
So, Dr.
And
Pete, before we get
yeah,
into this, I think it would be better for the audience if you can talk a little bit about the polyol pathway and that fructose uric acid metabolism. And you also mentioned that glucose plays a major role in that as well, because, when it comes to the fructose, people, they just say like it doesn't, trigger the insulin stimulation. So could you please talk more about polyol pathway and how the uric acid is being produced inside the body?
okay, so first, if we just focus on the fructose aspect of this, when someone takes in fructose, if we just talk about the liver effect, now we do have to be careful here because most of the fructose that you take in, so say somebody drinks a soda, like a Coca-Cola that has a lot of sugar in it, or they have a concentrated fruit juice, for example, which has a lot of fructose in it. Recognize that most of the fructose that's coming in there is converted to glucose by our intestine. But when the amount of fructose coming in is super highly concentrated, there's a certain fraction of that fructose that is going to reach the liver. And fructose, along with alcohol, I know stuff is really complex, but along with alcohol is a profound activator of fructose uric acid metabolism. And what happens there when fructose enters the liver is that there is an acute decline in our energy, right? And to keep things as simple as possible, we'll just call it an acute decline in ATP. And when that happens, you see an acute rise in uric acid. And what is on an intracellular basis, all right, inside the liver. And what isn't uniformly understood is that uric acid is, in fact, a signaling molecule. So when you have that is responsive to any biochemical process that happens in any of the tissues where this occurs, to acute changes in ATP. So in the liver, you have this fructose effect. ATP declines dramatically along with phosphate, by the way. You have this acute rise in uric acid, which is a signaling molecule, which downstream drives mitochondrial dysfunction and a shift to the synthesis of fat directly in the liver and also glycogen, but stimulates directly systemic inflammation, an alteration in nitric oxide signaling, which downstream affects significantly not only blood pressure, but it significantly alters the metabolism of the cellular layer that lines our arteries, along with other effects. So the uric acid effect is very important to the metabolic effects that are driving most, if not all, metabolic diseases. And because of the mitochondrial dysfunction that happens, if you do this decade after decade after decade, This actually, in the long term, leads to mitochondrial insufficient and the cancer condition. And then one aspect of this that I think probably none of the other influencers are even going to discuss, when this happens in the liver, this drives substantial peripheral effects. So the liver sends signaling along with uric acid and lactate into the peripheral circulation. So we're talking about changes in regulation, metabolic regulation at the level of muscle, adipose tissue, and perhaps very significantly in the brain.
So basically what you mean to say is like fructose, it actively reduces the ATP levels in the cells. It affects the mitochondria and then the body enters the storage mode. Is it something like body, whatsoever you are feeding, body is not able to produce active energy and it's producing the storage mode?
Yes.
Okay.
And when we come back and we ask the question, okay, so because that argument I just gave you makes it sound like, okay, so we don't have an issue with glucose then, but we do, because in the liver and all human tissues where this has been looked for rigorously, we have the enzyme aldose reductase. Now, in the liver, when you look at the historical biochemical data, I'm not talking about any of the observational stuff, no associational, none of that external sort of like literature that is not so good. When you go back and you look at specific biochemical literature that was published, peer-reviewed, high-quality work, starting with Warburg, and then Hans Krebs and Veitch, and then coming forward into the 60s through the 80s, with the literature that was published there, right up to the nutritional guidelines when they were published in the early 80s, where the kind of literature I'm talking about was then defunded over the following decades. What you see is that aldose reductase, along with the primary protein that activates the processing of glucose, both of these are glucokinase, both very similar proteins. When you look at them biochemically, their evolutionary origin came before the more complex protein and enzyme systems that we have in animal cells, right? The ones like hemoglobin that are all multi-subunited and all of that. These two proteins, glucokinase and aldose reductase, are actually activated biochemically, very strong evidence for this, under very similar incoming glucose conditions, which the activation is happening somewhere close to 90 mg per deciliter of intracellular glucose. And the reason that, and it's around this value, the reason that I have to hedge on this is because we don't have enough biochemical studies, in vitro studies, and in vivo studies that have been done on this. It hasn't been pushed on hard enough. But here's the thing. We know that aldose reductase, which is the first rate-limiting enzyme responsible for opening the polyol pathway, which is a two-step pathway that produces fructose directly in the liver from glucose.
Okay.
And once we activate aldose reductase, 30% of the glucose that's in the liver is now being pushed directly into making endogenous fructose. And endogenous fructose, at the point where you're making that, the fructose uric acid pathway is blown wide open.
So basically, if a person is a type 2 diabetic, okay,
Yeah.
and he is consuming a lot of fructose, so all that fructose will be then turned into the glucose or the polyol pathway, because you mentioned those enzymes get activated after a certain level of the blood glucose.
Yes.
So if a person is already insulin resistance, so that can elevate the uric acid levels?
Okay, so hold on, because the insulin resistance issue is is what I call a macroscopic control that is in fact involved in all this. But where does the insulin resistance come from?
Yeah.
Well, when you activate glucose, and so I'm going to call it by what I think is more correct. When you activate glucose, fructose, uric acid metabolism, the type 2 diabetic, so in all of this, let me just back up 1 minute. You have two things going on here.
Mm-hmm.
Activating aldose reductase, right? So that you're feeding glucose directly into fructose uric acid metabolism. But when you do this, and when you have incoming fructose along with the elevated glucose, you also increase the expression of a number of different enzymes. One of those crucial to this is aldose reductase. So not only are you activating it, but you're making more of it. So basically you're just kicking the soccer ball of glucose harder into the formation of endogenous fructose, which literally like a funnel is falling into this pathway. You have accelerated glycolysis, you have the elevation of lactate, you have all those things. And then what I said was, and this is super important, you have a tremendous amount of liver signaling that's going downstream. You have the direction of hyperinsulinemia into the peripheral tissues. You have insulin resistant signaling that also is being pushed downstream, which is driving. I think everyone, like if you guys are talking to Bickman, everyone agrees that hyperinsulinemia that's getting pushed into the peripheral tissues is driving insulin resistance. So there's a direct connection in regard to very strong associational evidence here that when you activate these pathways in the liver, you're driving hyperinsulinemia and insulin resistance downstream. So all of that is part of this.
So is it a bidirectional relation? So the high uric acid in itself can cause the insulin resistance that you mentioned. And once you are insulin resistance, you activate that polyol pathway. So it's like bidirectional. So it's a vicious cycle that person gets into.
Okay, so you raise now the issue of hyperinsulinemia. So I think that hyperinsulinemia and gout is a window into the soul of chronic metabolic disease. The only difference between, in my view, the only difference between somebody who's hyperinsulinemic or who has chronically high uric acid. So if you look at a type 2 diabetic and you ask the question, do they have high uric acid or not? Well, a lot of them don't. The distinction is now something that you have to take into consideration between a diabetic that has high uric acid and a diabetic that does not, because the distinction has to do with what's happening to the uric acid in the kidney. And for people that are hyperuricemic, high uric acid chronically, they have an excretion defect, which is genetically inherent in our population. So when I say our, I mean people like me that are hyperuricemic and also a gout sufferer, right? But the non-gout sufferers, they still have the excretion defect. Now, in studying those guys, what can we learn about these biochemical pathways that I believe that these central mechanisms that are leading to this whole constellation options, if you will, of a chronic metabolic disease that someone is going to end up with? Well, there have been a ton of both in vitro, in vivo, and mouse studies, for example, animal studies, and I think even some human clinical studies that show unequivocally that when uric acid is running high, it can diffuse into the tissue of interest, and it can elicit the same intracellular effects as if it was produced in that organ. We know this for sure with kidneys. You can diffuse uric acid into a kidney, and you're going to get out all of the pathological conditions that eventually lead to chronic kidney disease. And when you look critically, for example, at the gout data, you see the same phenomenon. We know that you can diffuse uric acid into a chondrocyte, the biologically important cell that is responsible for maintaining the health of our joints. And you see the same intracellular oxidative stress and so on, as if the uric acid was produced intracellularly. And also, this is important to mention. We know when you look at the chondrocyte and you go back into the high quality biochemical data that has been published in the past, that uric acid is in fact produced in the chondrocytes of gouty subjects. And there's even modern data on this. There's a group in New Zealand. The senior researcher there is Anthony Kettle. And I want to mention these other guys who are involved in this: Rufus Turner, Lisa Stamp, and Flavia Miotti, the Miotti papers. They have shown also with very good specific biochemical research that uric acid is in fact produced by chondrocytes. So every tissue where you can identify significant production of uric acid when you change the phosphorylation potential. So when you let ATP fall involved in these kinds of biochemical processes, when you diffuse uric acid in, you can have over the long term the same kind of pathological effects that you see when it's being produced directly in the tissue. So hyperuricemic people, of which we've got approximately 43 million in the US, and India has a bigger population. So I would imagine that your hyperuricemic population there is going to even be bigger than ours. And then we have a little bit over 4% of that who are also gout sufferers. So I imagine that you have this going on in India too, in regard to lifestyle.
So, Dr. Pete, you mentioned like uric acid is being produced inside the chondrocytes. So it's an intracellular uric acid production. But when we see like so many people here, like they have gout, but their serum uric acid level is normal. So is it something like the intracellular uric acid is causing that gout?
So let me try to do this in one sentence that's going to sum this up. We know from the research that came from the 60s, the 70s, and the 80s, and then very recently from the Kettle studies, that the intracellular uric acid is involved in this because you have this elevation of systemic inflammation that happens in the chondrocyte. We know from the Kettle work and this previous research that uric acid is diffused out into the extracellular matrix of the synovium. And the Kettle data is very interesting because it suggests that the uric acid that is extracellular, which is pushed because it's rising to a higher concentration intracellularly, it comes out into the synovium. And out there, it's involved in an oxidation reduction network with at least 3 other factors: ascorbate, a protein like or is myeloperoxidase, and a urate radical. And when the concentration of uric acid in that redox circuit rises to a high enough concentration, then you can get the formation of seed crystals. And the track into a gout flare requires two steps, because once those seed crystals form, if they get modulated quick enough, in other words, converted back into soluble uric acid, then you cannot advance to the second step, which involves, this flood of neutrophils and macrophages and then get the flare. So the crux, or the most important aspect of gout, is what's happening in step one. And if the seed crystals rise to some threshold which is unknown, then the processing of those crystals kinetically will not happen at a high enough rate. And you advance to this next step where the macrophages are overwhelmed. They start to break apart literally, and you get this cascade with the neutrophils into the actual flare, which is super hot. There's a higher temperature and a pain threshold, which most gout sufferers are going to say is a 10 on the scale of 1 to 10.
So, Dr. Pete, my next question is like, how important is the early diagnosis of the elevated levels of the uric acid? As you mentioned that it can lead to the various degenerative diseases. But when you say like the high uric acid level, what levels of the uric acid you consider high?
So that is a really good question, and well, you asked more than one. But here's the thing, right? We don't really know what normal uric acid is in a human. We know, if you look back at early historical data in the early 1900s, the average uric acid in our population, both men and women, was around 4 mg per deciliter, around 4 mg per deciliter. And now that average is around 6 mg per deciliter. In our space, there's a lot of hand waving in the low carb community because we know uric acid rises, right? Or both on carnivore and in a low carb environment. And these guys will argue, okay, well, when you reach keto adaptation, the uric acid comes back down to 6 mg per deciliter. That's the average in our population, so everything's okay. But the truth of the matter is, because uric acid historically anymore is not measured, it might not have ever been measured when in our children and then young adults and so on coming forward, we don't know when the hyperuricemia, is it there from the beginning? Is this a genetic thing that in part of the population, this is the way it is? We don't really have an answer to that. And quite frankly, when I raised the issue about the fact that in the early 1900s, the average was around 4 mg per deciliter, and now it's 6, that usually causes whoever I'm talking to go silent. And honestly, when you look at the data that's out there, which is almost completely associational and whatnot, it's multifactorial. You can't say, for example, it's because we're eating more protein now, because honestly, eating meat hasn't changed that much. I suspect it's a matter of the rise of sugar consumption, processed food, along with the rise of alcohol, and while meat. Okay, so be really careful about putting blame on meat, because eating protein is only a small contributing factor to elevated uric acid. The majority of the effect is a sugar effect and an alcohol effect, right? If you take in a high quantity of sugar with both elevated glucose and or elevated fructose and have alcoholic drinks while you're doing this, so if you're looking at a plate of food that's hyperglycemic, lots of potatoes and rice and a sugary dessert, maybe you're having an alcoholic drink like a daiquiri would fall into this category that also has sugar in it and so on, you are going to be overproducing uric acid in your liver, whether you want to or not, along with lactate effect that it's pushed into the peripheral circulation and all of the things that we were talking about, the downstream signaling in regard to insulin and lactate signaling actually because uric acid and lactate, along with beta hydroxybutyrate and acetate, all of these small molecules, evolutionarily speaking, were involved in the control of our metabolic pathways a long time, millions of years before you had the macroscopic controls like cortisol, insulin, and all of these, this next layer of regulation.
So, Dr. Pete, my follow-up question for this, like you mentioned, the uric acid production, it depends on the consumption of your fructose, alcohol, and hyperglycemia. But what about the salt? Like when the high carb diet, the standard American diet, is in combination with the salt, is there any relation that it can activate the polyol pathway?
Yes. So the activation of the polyol pathways is affected, right? You have an increase in expression of that enzyme under conditions of hyperglycemia, under conditions of alcohol, under conditions of sugar. And when I say that, I mean it generally. So however you're getting it in there, table sugar with glucose, fructose, and so on, and the elevation of salt, right? And hypoxia. This is why the sleep apnea problem is tied into driving chronic metabolic diseases and gout, because hypoxia activates and increases the expression of aldose reductase, which then increases our sensitivity to incoming glucose in the liver. And this is why, and I know where you're going with this. So one thing, I know that we're running out of time here. So very quickly, when you shift to a low carb diet or carnivore, because we're bringing the insulin expression down, you see a change in relationship with incoming salt at the level of kidney. So we have to really be careful here because we're talking about different physiological states. So somebody on the standard American diet, in general, right? And part of this comment is coming from 7 years of coaching really sick people. But basically, they are just eating a plate that's hyperglycemic with a lot of sugar coming in and alcohol, usually for the adults. A lot of times, they're also eating a high quantity of processed food. And processed food, what does it have in it? Normally, it's going to be hyperglycemic. There's going to be sugar in there. And they have altered the salt and also other things that we haven't even talked about here, like glutamate and compounds related to glutamate, along with certain types of fats. And I didn't even get to the part about glutamate and the fats because they also are contributing to opening in a very big way this central metabolic pathway that's driving downstream chronic metabolic disease. Now, when we switch to a low carb environment and or carnivore, everyone knows you've got to modulate the salt. And that's because when you pull the insulin off, now we're pushing during the keto adaptation phase, we're pushing salt out into the urine. And to avoid the keto flu, I'm just going to call it that, you've got to make sure to be getting the right amount of salt. You have to be taking in more of it. But eventually, in the case, this is looking downstream, right? At guys like me, perhaps you, I'm not sure where you are in the journey, right? But I've been doing this for 7 years. I've been eating this way religiously. And I don't mean willy-nilly. I mean, I took my biomarkers this morning before I talked with you because I'm still trying to, coming back into the science, understand what's going on here. And because of my work in the early, looking at the actual science that was published by Warburg, by Hans Krebs and Veitch, I know that the standard American person eating the standard American diet and compared to the ketogenic person, by comparison to a carnivore person, that these are all defined physiological states. And our processing of energy is dependent on the ions that are related to how our cellular systems are able to process ATP, to keep the ATP levels, the proportionalities of our energy correct and in a zone where our tissues can function. And the fluctuation of ATP compared to its other to its other related molecules is dependent on the sodium, the potassium, the magnesium, the calcium, uh, and other ions that I can't, like bicarbonate that I can't think of right now. And I know I'm going into the weeds with this, but this is so super important to us because these physiological states are driven by these mechanisms. And even though low carb and carnivore overlap significantly, right, we have similarities, much more similar than the standard American diet. Your relationship with salt will change the longer you're in a low carb environment, right? This idea that keto adaptation happens and then you're done is not correct. The longer you're in this environment, the more our physiological state in regard to how we respond to incoming nutrients is going to become more formatted in a way, to think of a term with that. All right, and I better pause there.
Yeah, so Dr. Pete, really, so I'm so excited about this topic now after listening to your session. So one more thing I would like to ask. I know that you are a very active researcher and you are studying actively more on the uric acid. I think if you have certain latest studies or interventions about the uric acid that you would like to talk.
Okay, so people ask me all the time that are hyperuricemic, what do I do? I'm on this low carb and I'm seeing these extreme values. Okay, so step number one, before you think of any other interventions, it's to get stable with your metabolism. And what do I mean by that? You need to get keto adapted, number one. If you've gone onto a ketogenic diet, you need to get keto adapted. And in this space, a lot of the investigators or whatnot are going to say this is around 3 months. But for old people like me, I'm 66 years old. I know from experience and working with a lot of clients that keto adaptation in older people can be significantly longer. I would say you're probably looking at a year, maybe even. So you need to get stable in the lifestyle and let your uric acid come to a level which is normal for you. And if you are hyperuricemic at that point, then you have a decision to make. And the big thing is that understand that we really, when it boils down to it, push comes to shove, we don't know if hyperuricemia is safe or not. I'll take anyone that wants to argue about that to task. We really don't know. The only thing that we've got are observational and associational studies, and a lot of them point to that you're at a higher cardiovascular risk for some type of an event. So here's what I tell everyone who asks me this question. At that point, then, that you're still hyperuricemic, either on the carnivore diet or on a ketogenic diet, and I mean ketogenic now, so 50 grams of total carbs or less per day, then you've got a personal decision to make. Do I go into urate lowering or not? And if you decide you're going to do it, I think the best way, and there's a lot of supplemental things you can do and whatnot, but I believe you should just go for the throat and consider allopurinol. But you need to titrate onto that. Going to your medical provider and saying, okay, I'm hyperuricemic, I want to go on allopurinol, that doctor might say to you, okay, we're going to put you on 600 mgs allopurinol per day or something like that. And that is not the way to do it. What you want to do is start low and titrate yourself up over several months. And what do you want to settle at for a concentration of uric acid? I believe you go with the concentration of uric acid that people were expressing in the early 1900s, because that was before the advent of all of this sugar and alcohol evenly across our population, which is about between 4 and 5 mg per deciliter. That's the target. Now, you also, it turns out, can attack the excretion phenomenon at the level of the kidney, and you do that with glycine, right? And there's been really good, high-quality studies done that have shown in a dosage-dependent manner that if you increase the amount of glycine in your diet, that will directly cause uric acid lowering, because it increases the excretion of uric acid at the level of the kidney. And I've experimented directly with all of this methodology, and you can use both the glycine and the allopurinol together. And so what I recommend, if you're going to attack the hyperuricemia, that you very carefully titrate up the allopurinol over a matter of months. Your target is around 4 to 5 mg per deciliter of uric acid, and you couple that with glycine and find where your equilibrium best works. And then I guess people are probably asking the question
Dr.
right now.
Pete, I'm really sorry to stop you, but because we are already running out of time, so it's
okay.
so yeah.
You can measure your uric acid with a meter. All right, that's it.
So okay, so before we close the session, I would like to call Shashiji and Anup Singh.
I'm sorry, say that again.
Yeah. Peter, thank you. I just came to thank you for coming in this conference. And I always had your mind that you are an authority on uric acid and gout. Since I saw multiple presentations, especially Nutrition Network, I always had you in mind, and I was just waiting for a particular event. So thank you very much, and we will meet again. Probably we'll invite you in a podcast where we can have extensive discussion, and Jasmeet will host you again. Anup.
Awesome. I would love to do that. Thank you.
Thank you, Dr. Peter. And before I sign off, I think I will have to listen to this episode at least 10 times to get hold of everything. It was such a brilliant one that you can take it as I would say not less than 10 times, because every minute you have been talking science and every minute you've been talking data. Thank you. Thank you very much. And
thank you so much, Dr. Pete.
Yeah, we'll have you on a podcast. Yeah, we'll have you on a podcast for sure.
Yeah.
Awesome. I'd love to do that anytime.
Thank you.
Thanks you guys for having me.